It is a pleasure for me today to talk about a quite important topic, which is gut microbiota dysbiosis, and in a way how we can measure it, how we can profile it. I will start with a really simple question, which to all of you, which is who has not ever experienced some of the following symptoms: stomach pain, gas, constipation, diarrhea, fatigue, brain fog, concentration difficulties, anxiety, and so on. So a lot of different symptoms. Then the answer, usually, it might be in your gut. And that's why we say usually the gut is the second brain or a unknown organ. And a lot of focus have been coming into the research based on the fundamental research and clinical research into this hidden organ. So when we talk about dysbiosis, first of all, in the human gut microbiota, we always hear about two terminologies, two terms, which are: the first one is microbiota, and the second one is microbiome. The microbiota is what we call the microorganism by the type, which are living in a specific environment. That means the individuals. It's a community of fungi, viruses, phages, bacteria, everybody living in your gut. On the other hand, you have the microbiome, which are the same organism, where we include the genes, meaning the individuals and their functions. And within this community, the majority of these microorganisms are the bacteria. And the bacteria, there are 6 different phyla up to date, and approximately 90% of these bacteria belong to the Firmicutes and Bacteroides phyla, and the 10% belong to the 10 others. That's basically the basis. A healthy gut, we cannot talk about dysbiosis before knowing what is a healthy gut. A healthy gut is a fine balance of the gut bacteria, which means the composition of the gut microbiota remains quasi stable and unique during the adult life, and has a fingerprint. However, in all of us, it provides essential biological functions, as we heard a little bit earlier, and then we will hear. And that means protection and nutrition. And as well, we have some common core bacteria species. And this fine balance, it's between the beneficial bacteria, which we call the commensal bacteria, and the conditional and pathogenic bacteria and pathogenic bacteria. So we have to have the majority of beneficial bacteria versus the others. And a healthy gut, it's not only about the community, a good representation of healthy gut community and organism, as I said, with bacteria, bacteriophages, and so on, but it's as well about physiological responses. And usually we categorize them into 4 different functions. One is about anti-inflammatory effects, about immunology, and all about immunology. And the second one is about the prevention of pathogen colonization, so really about the protection, as we heard before. A third one is about the digestion, because those bacteria will facilitate the digestion of complex carbohydrates and complex fibers. And the fourth one is about as well about nutrition, but it's about the production of nutrients and the production of vitamins and beneficial metabolites like single chain fatty acids, lactate, and so on. And all this community lives together quite nicely, but they are in really symbiotic relationship with us, with the host. That means us. And then this can be disturbed. And that's what we call gut dysbiosis. It's what we call an alteration or disturbance, an imbalanced state of this gut microbiota community, which is associated with unhealthy outcomes, either a disease, symptoms, and so on, which I will deepen a little bit more now. How can it happen from eubiosis to dysbiosis? And that it's a mix of 2 different factors. That's where I sum up here just a few ones. It can be some individual factors, intrinsic factors like genetics, like the age, like the illnesses and the injuries of that we have, versus the individual environments, especially the medicines. We know that when we are taking antibiotics, that's really creating a dysbiosis state because they are killing some bacteria species which are in our gut, as well as some infections that we know with the last 2 years with the COVID-19, how it has affected, will affect the gut dysbiosis, the gut balance, the lifestyle, what we are eating, it could be diet, it could be smoking, it could be activities, it could be a lot of different factors, and the pollution as well, what we are breathing and so on. So a lot of different factors. And the common features of gut dysbiosis in terms of bacteria, it's like four. We categorize in four. In the first one, it's a significant change in the relative proportion of the major bacteria families. For example, you could see in this example, we have more green species than others. A second case is when there is a reduction or total loss of some gut commensal microorganisms, some beneficial bacteria. A third one is a decrease in the diversity. We have less bacteria. And the fourth one is when there is some potential pathogenic microorganism which are taking over or growing. So all those four can happen alone, but usually they can happen as well all together. And there has been reported now some disease associated with intestinal dysbiosis. We know there are some traveler diarrheas, some antibiotic associated diarrhea. There are studies and results about it. You have a lot of different gastrointestinal disorders and gastrointestinal disease like irritable bowel syndrome, inflammatory bowel disease, gastroenteritis, gastrointestinal cancer like stomach or colon cancer, even if we know there's a dysbiosis state. Infantile colic, it could be between 20 to 25% in the children, and as well some metabolic disease like obesity. But the main question now, today, which is not yet answered, is it the dysbiosis and this imbalance in the microorganism in the bacterial community? Is it the cause or the consequence? And I know we don't have the answer now for the moment, but a lot is working on. But it doesn't stop at the gut, because this imbalance in the gut, which is really influencing the, I would say, the inflammation or anti-inflammatory process, because you have the pro-inflammatory, anti-inflammatory systemic responses. You have an incidence in these on the terms of dermatological conditions like acne, allergies, some metabolic conditions, as I mentioned before, obesity and diabetes, mental as well, anxiety disorders, neurological in autism. There is some reports about dysbiotic gut in autism individuals, and even now we think about neurodegenerative like Alzheimer's, like Parkinson, with this gut-brain axis. And recently, or I would say the last 2 years, we've seen the influence of the infectious with the COVID-19. I will now take like some examples. The gut dysbiosis and irritable bowel disease, the IBD. It is IBD, it's ulcerative colitis and Crohn's disease. It has an inappropriate inflammation in the intestine, as you could see in the pictures below. It's a series of relapse and remission affecting essentially young subjects, young individuals. It impaired a lot about the quality of life, and there is no curative treatment for the time being. Reports show that there is a change in the gut microbiota composition. We know there is a loss of diversity. If you remember, that was the case number 3. And there is a lot of stimulation about the inflammation. And when we look at the properties of the bacteria and their properties, we see that there is the pro-inflammatory bacteria are in majority versus the anti-inflammatory bacteria. And that's the dysbiosis. It's thought now to play a role in this disease. Another example, because it's about COVID-19, and that's another axis, is the gut-lung axis, where it was reported that 50% of the COVID-19 patients developed gastrointestinal symptoms. That's, we know it has been reported a lot. And if we look at the gut microbiota composition, there is a loss, I would say less diversity, depletion of beneficial commensals, and an increasing of opportunistic and common pathogens. And then the gut dysbiosis here has been thought maybe to be a predisposition to the infection of COVID-19. If you had a dysbiotic gut from before, maybe you were more predisposed to COVID-19. As well, a gut dysbiosis state was linked maybe to disease severity and such as well poor prognosis. In terms of physiopathology, we've seen there have been seen a lot of inflammatory reaction. This leaky gut phenomenon has increased. The spillage of bacterial products and toxins has been increased. So everything created this inflammation situation and maybe this cytokine storm in young individuals, as it was reported. But when there is this dysbiosis state, it can be the recovery. It's geographical and individual dependent. It can take 1 week and it can take up to 6 months. That was in the Chinese paper was reported. So it's what we call the long-term effect of the COVID-19 on the gut. But then my question now is, how can we clinically measure dysbiosis? Because we have to find some, because so far it was some research, what has been reported, but it was how to identify microbial markers for diagnostic purposes. And once it's done, we have to build a really robust and reliable diagnostic tool, which should be used for routine, for in any lab, in the healthcare professionals, which should be standardized, so really robust, and should be clinically and technically validated. So that's the main parameters. And I would like to introduce you to the GA-map Dysbiosis test, which has been developed by Genetic Analysis, where I work today, which was the first documented and validated CE-IVD fecal test, which aim to identify and characterize dysbiosis in adults, and it is based on determining a deviation from normobiosis. This GA-map, when can you use it? Actually, you can use it to determine if a patient is non-dysbiotic or dysbiotic. You can use it to establish a gut microbiota profile in this patient, and you can use it to monitor dysbiosis during the treatment. When we developed our test, we focused on IBS patients, IBD patients, symptomatic non-IBD patients, meaning those patients had IBD symptoms but had colonoscopy negative, and some FGID patients, so everything about gastrointestinal disorders patients. So with the COVID-19, you all heard about the PCR, but PCR in this for COVID-19 and SARS-CoV-2 detection is I would say is focusing on one target. In our case, this GA-map technology is a PCR technology base, which has been much more enhanced, to make it simple, much more developed with bioinformatic tools at the end. And it is a multiplex detection where we are using 48 bacterial DNA probes covering the 6 phyla, as I explained, as I remember the beginning of my talk about the different taxonomy levels, focusing on the bacterial 16S RNA gene. These probes, for today we have 48, they are specific probes and they are dysbiosis specific group probes. They were based on the literature research, they were based on the discussion with opinion leaders and gastroenterologists, they were based on clinical data which we had. So it has been really intensive research and clinical validation back of it. We had developed an in-house algorithm that combined all this information for these 48 probes and the different data we have from these probes to make it really simple and really to everybody in order to read what is the dysbiosis test. It is a 5-point scale going from 1 to 5. 1 and 2 means that the patient samples is normal biotic. 3, 4, 5 means the patient sample is dysbiotic, with going from mild dysbiotic to severely dysbiotic. And that indicates how much this bacterial profile of the sample deviates from this normal and healthy reference. We have a standardized patient report because everything has to be routinely used and easy to use, which contain 3 different parts. The first one, as I mentioned, the dysbiosis index with 1 to 5. A second part about some imbalance into functional profiles, which I will deepen a little bit after. And the third part about the abundance table of these 48 probes in comparison to a reference range population, how those probes differ in a way. The functional imbalance, because you remember I said dysbiosis is not only about the microorganism, but it could be as well about the functionality. Is here we have selected some group of bacteria which are butyrate producing. So we see if they are in balance or in imbalance. We have indication about these gut mucosa protective bacteria, the gut health marker, Faecalibacterium plantnitzii, which is a really known gut health marker and the most abundant bacteria in your gut. Some indication about pro-inflammatory bacteria, Proteobacteria shingella, and as well some diversity. So a question I had often when I talk about dysbiosis is, can we restore an imbalanced gut? Can we restore, can we go from dysbiosis to eubiosis? Interesting question, which is in a way dependent of the symptoms, dependent of the disease, dependent of the patient, but dependent as well of the biology of the dysbiosis, because it's called a resilient state, actually. And I would say it's not as simple as bringing good bacteria or replacing the missing bacteria or replacing the bad ones, because we have to have a global eye, a global view. And the idea is more to influence the gut microbiota to help it to function smoothly and thereby improving the whole host health. There are different ways to do it. You can change your diet. You can use probiotics. You can use prebiotics. I'm just listing 4 of them. There are other ways. Or you can do a more invasive, I would say, approach, which is fecal transplantation. But I would say how ethic our genetic analysis is that when we run an analysis of a fecal analysis, we are not sending the sample report directly to the patient. We always work with healthcare professionals, and we stick to that any intervention should be undergone and should be decided by the healthcare professionals, by the lab, by the doctors, because those are the only ones who can decide which intervention should be chosen, which follow-up plan, which monitoring should be done, how often, and everything based on patient health history or dysbiotic state, and so on. Now I'm talking about another aspect of this dysbiosis test is what is the clinical utility? Because now it was on the routine use and for your patient, for in the lab, but when you are doing a little bit some research or clinical research, then I would recommend you to check how key publication from 2015 explaining, which is called Deviation in Human Gut Microbiota, a novel diagnostic test for determining dysbiosis in patients with IBS and IBD, where it is explained on the basis of how reference range population, which shows a really good separation of the normal to patient samples, where as well it is explained the technical verification and clinical validation, where we see, for example, the repeatability and reproducibility of how test is less than 2%. And all this has contributed to the CE-IVD marking to our test. Some results, we saw that in a healthy population, 60% of the individual have a mild dysbiosis, you see in the green. So as you could see. And then in patients with IBS and IBD, we have measured that between 70 to 80% have a microbiota profile that goes from have dysbiotic. IBS patients have more mild dysbiosis. IBD patients where the inflammation is higher are more towards 4 or towards 5, really severely dysbiotic. Another data which we found, it's this kind of bacteria, predominant bacteria contributing to dysbiosis. For example, in IBS patient, it's more an increase of signal of some bacteria like Proteobacteria, actinobacteria. You could see on your left, on the left hand side graph, the red bars are higher than the gray one, which are dysbiotic versus non-dysbiotic. Whereas in IBD, it's a mix of increased and decreased signal. For example, Faecalibacterium prausnitzii, the signal is decreasing, whereas Shigella and Escherichia coli is increasing. So the GA-map dysbiosis, if you on the clinical aspect, we have per today 40 scientific publications with an impact factor of 4.5 if you're interested. We have worked towards with research groups into inflammatory bowel syndromes, ulcerative colitis, Crohn's disease, arthritis, obesity, and many other symptoms as well. And we worked with, for example, and they worked how partners with clinical nutrition, microbiota profiling, microbiota monitoring during a treatment. So and then we have discussion about it. And now I would like to show you like 3 case studies, because I cannot show you the 40 publication, but 3 case studies which could be relevant for dysbiosis. The first one is about functional gastrointestinal disorders, FGID, from a publication published last year in 2021, where if you check on the green boxes, it is about the dysbiosis was more frequent in subjects which were positive for anti-cytolethal distancing toxin B antibodies, and in IBS-C patients, so patients which have more constipation. Here it was the highlight was for you to know the anti-CDTB and anti-vinculin antibodies which are measured here are thought or known to be some biomarker to identify post-infectious IBS, and it was to correlate with dysbiosis and microbiota profiling. And at the same time, so in this IBS-C patient and anti-CDTB positive, it has been seen an increased amount of opportunistic and pro-inflammatory bacteria and a reduction into gut protective bacteria. The second case study is about FMT treatment in IBS patient, FMT fecal material transplantation in IBS patients. Studies which we started some time ago by a group in Norway, and has been really a lot talked into international conference because of the really excellent results out of it. First of all, how test was used to test the gut microbiota of a well-defined super donor, where we showed this super donor has a DI score, a dysbiosis index score of 1, normobiotic, with an abundance of bacteria belonging to the streptococcus, belonging to Firmicutes, like Streptococcus, Dorothea, Lactobacillus, and Ruminococcus. As well, how test was used on the GS score. The GA-map was used for the patient on the patient's gut microbiota, was analyzed using how test. Here are the results after 1 month. So the patients received either 30 grams, either 60 grams of fecal material. And there is a significant decrease of the GI score in the patient with 60 grams. So that means there tends to be less dysbiotic. And the 30 grams is not significant after 1 month, but it became significant after 1 year, indicating that the intestinal bacterial diversity had improved after 1 year. And generally, we see it has a tendency that in the patient, the bacterial community tends to be like the donor. We've done some profiling, the analysis of the changes of the intestinal bacterial profile following the transplantation, where there are significant changes in the 2 groups and not in the placebo, which was really good. And generally, after 1 year, there is a really good changes in the GI score, dysbiosis index. So very good into fecal bacterial profile and into single chain fatty acids as well. That's was reported. The gut microbiota profile changes after a year in the patient. Here we had a mix of 30 grams and 60 gram patients. So all the patients which were included in these clinical studies, you can see some bacteria have had increased, some other had decreased. The one who decreased, the proteobacteria, the Firmicutes, a lot of proteobacteria had decreased, and most of the Bacteroides had increased. And currently, there is recently some focus about alistipes, which it seems to play maybe a central role in the improvement seen after the FMT. It has to be, it needs to be shown, it needs to be investigated further, but it might be more into male than female in a way. The last study is about IBS, crossover intervention studies, where it was about 20 IBS patients, which followed a low food map diet in 3 weeks, followed by a high food map diet in 3 weeks. Here we observed a single bacterial changes. That if we, I took the example of Faecalibacterium proudnitzii and Bifidobacterium, where after the high food map diet, for example, for Faecalibacterium proudnitzii is increasing or going back to the baseline, whereas for the Bifidobacterium, the low food map diet is decreasing. So this is this kind of options we can make with our test in the clinical settings. So if I sum up the clinical utility of the GA-map Dysbiosis test, it's about identification, characterization, and prediction. We talked about microbiota profiling. We can work on treatment response. We can work on disease subtyping, especially in IBS, treatment escalation. When do you need to stop the treatment? Has been worked on it, and the disease severity course. So those are mainly the clinical aspects of our product. But I was talking, I said, we are genetic analysis. It is true. So where does it come from? We are Norwegian, as Lara said, since 29. We have worked on in the field, in the microbiome field since 29. The GA-map technology is a patented probe-based technology with 40 peer-reviewed publications used in 50 clinical trials. Our test, the dysbiosis test, is used as a routine test by local and global molecular diagnostic labs, both in Europe and in US. Sometimes it is under other names, but we know that our technology and test is used there. We have international collaboration with pharma companies, but with academia, and we as well have new biomarker development projects to work on other kind of disease area. So I hope that I have introduced you to the GA-map dysbiosis, to the dysbiosis word, and how we can measure it, how we can use it or profile it in the routine aspect and as well in the clinical aspect. So thank you for your attention, and I wish you a nice day.